Welcome back to the Flagship studio here at the JP Morgan Healthcare Conference. We are rounding out day 3 with Eric Shaff, the CEO of Seres. Eric, thanks for being here.
Thanks for having me.
Absolutely. So Seres has established itself as the company harnessing the microbiome to develop drugs to fight some pretty serious infections. And you've shown that this can work with Vowst. How are you taking those capabilities and the systems and the platform that you've built for Vowst and applying it in a new direction?
Well, Seres was founded about 10 years ago with a series of what if questions, right? And there was this early proof of concept that fecal transplants could provide a signal that you could interdict into someone's microbiome and have an impact on a serious disease like C. diff. And the founders of Seres said, what if you could elevate the science and understand how the microbes are interacting with each other and human tissue? What if you could produce a product under GMP conditions? What if you could elevate the science and study patients under rigorous FDA oversight? And hugely important, what if you could make it oral, right? Yep. So we've gone through the journey with what was then Sera 109, what is now Vowst. And to answer your question, we are changing and saving lives each day with Vowst. And Vowst is not an aspiration, it's here today, and it's changing the standard of care. We are absolutely sure that the lessons that we've learned through our journey with Vowst are applicable to the future of our technology, and that starts with Sera 155, which is in development for patients undergoing allo-HSCT. So the lessons that we learned, the technology, the scientific know-how, the manufacturing, the relationship with FDA, all of those are applicable as we think about the journey forward with 155 and beyond.
So you just introduced a new term, allo-HSC. What is that? And then how is Sera 155 designed to help those patients?
Yeah, so if we think about preventing infections from medically vulnerable patient populations, certainly patients that are undergoing multiple rounds of C. diff fit that bill. And we've thought about what are the adjacencies from those conditions where they're medically vulnerable patients who are susceptible to pathogens. So think about not just the pathogen itself, but the system and the susceptibility of the system to pathogens. And for us, cancer patients undergoing allo-HSCT fit that bill. So these are patients that are really hammered with antibiotic therapy, with chemotherapy. They're vulnerable to bloodstream infections, which is a significant problem, growing actually with the change of standard of care with the introduction of PT-Psy. But specifically, what physicians and patients care about is being derailed from their underlying cancer treatment, and bloodstream infections do that. So the idea behind Sera 155 is really a couple of fold. One is to prevent the overgrowth of pathogens, similar to what we did with Vowst, right? The second is to protect the mucosal barrier from the translocation of pathogens from the gut to the bloodstream. And the third is to enhance systemic inflammation. And so those different mechanisms together were behind the design of Sera 155, and we recently completed a phase 1b study which we're really excited about.
Yeah, I was just going to go there. It sounds like you're starting to see early signs that this is working. Tell us a little bit more about the data from the tail end of last year.
Yeah, so a 1B study evaluating Sera 155, we had 2 cohorts. The first cohort was open label. So because these patients are so medically compromised and vulnerable, the idea of dosing with high doses of bacteria, perhaps not a foregone conclusion that was okay. We got through that first cohort. It was great. We were happy with it. We saw signs of a reduction in the pathogen overgrowth that you might otherwise see in a reference cohort that we worked with Memorial Sloan Kettering over a number of years to establish. The second cohort was placebo-controlled, and we saw a number of really interesting signals. The primary endpoints were engraftment of our organisms, as well as, of course, safety. We were pleased with those results. But we also saw a significant reduction, actually a 77% reduction in bloodstream infections when you compared the placebo arm to the active arm. That's a big deal for physicians and for patients. We also saw a reduction in febrile neutropenia and a significant reduction in the amount of exposure to antibiotics. So for our KOLs, for folks we've spoken with, they think that this is an incredibly exciting result, and they're looking forward to the next steps for us.
Speaking of next steps, you have expanded on those initial phase 1b data with a new study that you'll present at a conference next week. Can you give us a very high-level view of what those data will be?
Yeah, so in addition to the initial clinical results, we actually just released that there's additional data that we've gotten from our pharmacology analyses, which suggests that we are, in fact, protecting gut barrier integrity the way that we had envisioned, and we'll have more to say in a couple of weeks when we present that data at Tandem.
We will stay tuned. So thinking about the year ahead then for this program, what are some of the next steps?
Yeah, so we were really pleased where at the end of last year, the FDA awarded us with breakthrough therapy designation.
Great.
To us, that signals that the FDA understands that there's a significant unmet need in the space. There's this exciting early data. There's a safety profile, which we've seen with this program, but with other programs as well. So we announced last week that we had submitted the briefing book for our proposal for the next study. We're hoping that that next study could be a pivotal study. Certainly, we'll hear back from the FDA as to their desires as to where we go. But we're doing the work to prepare for a next study, including working to establish sites, manufacturing, clinical material, and we're going to be ready to go with that next study, no matter what that study is.
Terrific. And are there other indications where you think you could take this product?
Yeah, so the initial indication is for allo-HSCT and the reduction of bloodstream infections. But with the mechanism of preventing infections, there are adjacencies that we think make sense. So in addition to allo-HSCT, think about auto-HSCT, think about other blood cancers. And certainly we have additional programs that we're contemplating in things like chronic liver disease, where again, the idea is to prevent infections in these medically vulnerable patient populations. And we're excited about where that goes.
Certainly the end goal here is to bring products to patients.
Yeah.
How do you think about the commercial opportunity for this program?
Well, we've done work with both payers and physicians, and what we hear back again and again is that physicians are really focused on getting patients through their transplants and ensuring that they survive. And derailments from the underlying treatment protocols through bloodstream infections, it's a serious problem. So commercially, we do think that the allo-HSCT indication, along with auto, creates blockbuster potential, and we're excited to move forward with that.
Okay. Eric, you have another day ahead of you as you will be presenting at the main JP Morgan Healthcare Conference tomorrow. What's your main message for investors?
Well, I think for us, our technology, it's not an aspiration to help patients somewhere down the road. It's not early evidence or hope. We are changing patients' lives with Vowst. And we absolutely think that that's just the beginning of the story for Seres and the beginning of the story for the microbiome. The technology that we've established, the lessons that we've learned, we absolutely think are applicable to our journey with 155 and beyond. So I think the message is we've got the right technology, the right team, the right science to change lives, and we're in the process of doing so.
Great. So a bedrock foundation for an exciting journey ahead. Thank you, Eric, for being here.
Thanks for having me. Appreciate it.